All right, so where we left off, I pointed out that there are times when you don't need to explain the origin of complexity as anything other than good old-fashioned natural selection on natural variation. Incidentally, if you're wondering, we talked about the evolution of the eye, starting with a little patch of light-sensitive cells. If you're wondering where a patch of light-sensitive cells could have come from, there are single-celled protists that are light-sensitive, that have various light-sensitive pigments. If you're wondering where the ability to send nerve impulses could have come from, there are single-celled protists that can actually pass action potentials. The simplest of multi-celled animals, the sponges, can actually pass action potentials from any cell to any other cell. So if you're wondering how cells could come to be sensitive to light, your retina cells are very specialized for being sensitive to light, but they're actually sensitive to other things. Don't do this, at least don't do this very hard, because if you do, you might damage your eyes. But if you squeeze your eyes or you press on them gently, the pressure in your eye will translate into spots of light on your retina. If you've ever squeezed your eyes really tightly and seen, flashes of light, that is because the cells in your retina are sensitive to other things besides light. They're very specialized for light, but other things can make them fire. So if you're wondering where that light sensitive spot came out, it's basically a working out of potentials that were present probably in very early eukaryote cells. Even single-celled critters can have light-sensitive pigments and can be sensitive to light. There's a big protist called blepharisma that's famous for this. And even single-celled protists can actually transmit action potentials. That kind of develops out of the basic machinery for keeping some atoms in a cell and pumping other atoms out, which is kind of basic to the idea of any cell. So even at the very molecular level, you're looking at natural selection acting on potentials that's already there. And to kind of jam on that, a French biochemist named Jacques Monod, probably most famous in biology as one of the team that discovered this thing that will make all of you shudder if you remember your genetics class. Anybody remember the lac operon? This set of genes in E. coli that regulates the production of lactose, of enzymes that break down lactose. Those enzymes are normally not transcribed and translated, but you've got the operator and the promoter sequence that act as controls. Anybody remember this? You've got the cap protein with cyclic AMP, and I don't remember the details anymore. But that basic idea of how gene expression is regulated was worked out in part by Jacques Monod. He also came to be kind of a philosopher of biology, and in a book he wrote called Chance and Necessity, he referred to the evolutionary process as a bricoleur. In French, bricoleur means more or less a tinkerer. Now, a tinkerer, okay, a designer like starts from square one and draws out a design, maybe beginning with an image in his or her mind, certainly later with blueprints or what have you, and then gets the parts and puts them together. The idea comes first. A tinkerer is the kind of person who will just go down and mess around with whatever happens to be on the shelf. will assemble parts and just kind of see what happens. I used to do a lot of field work in the Mojave Desert, working on fossils with a good friend of mine named Whitey Hagedorn. And I'm not very mechanical, but Whitey Hagedorn um, can fix anything. He is a master of the automobile. He has restored classic cars for fun and profit. And we got around in the desert with this incredible vehicle. He'd gone to a junkyard and bought the frame of a Jeep and just enough parts to get it home. And he's the kind of person who goes to junkyards for fun, looking for spare time projects. And over the next few years, as he had time and money, he'd go to junkyards, find things to put on this vehicle. And by the time I got to know him, it was this absolute Frankenstein monster um, of a Jeep. He called it the Heap. Um, he'd fitted it out in all kinds of ways. One that I vividly remember was he had a spare gas tank, which he'd salvaged from a boat and put into the cargo compartment right behind the passenger's head. I remember this vividly because I was the passenger, and if we had ever had a wreck, I would not be speaking to you now. I was risking fiery annihilation every time I went on a trip with him, but I went anyway because he was fun. Still is. Um, so he had this boat gas tank in the cargo compartment of the Jeep. He'd rigged a fuel line running from that tank to the main feed into the engine, which he controlled with a light switch mounted on the dashboard. So he could switch from the main tank to the reserve tank using this light switch. He had this roof rack on top that carried 2 tires. And that was very handy because driving over rocks in the desert, blowing a tire is always a problem. And, you don't want to get stranded out there. And usually we didn't, although there were a couple of, never mind, interesting times. The roof rack made the thing so top heavy, we were always liable to roll over in a light breeze. So driving it was kind of an adventure, but it got us into a lot of places and it almost got us out. It was an amazing vehicle. And I always quote that because Whitey was an example of a bricoleur. He did have some kind of goal in mind, something that would work well getting you onto, fossil hunting localities by rough desert back roads. But he didn't have a completely finished idea in mind. He put together what he had and added on to it using whatever was available at the time that would fit. And that's how we ended up with this Frankenstein monster with the enormous cast iron roof rack and the spare fuel tank operated by a light switch that gave new and frightening meaning to the term suicide seat, as I well remember, because I was usually in it, to a body that I think was basically held together with Bondo. Just an amazing car. And that's an example of how tinkerers work. You work with what's on hand rather than planning things from, the top down. You work from the bottom up, reusing what you have available for new functions. So that's what a tinkerer does. Jamming on that riff, Stephen Jay Gould and his colleague in South Africa, Elizabeth Verba, came up with a term, exaptation. We've been using the word adaptation to refer to the way in which species become over time better suited to the niches that they occupy. more likely to survive and prosper and increase their numbers in their niches. We know about adaptation. Gould and Verba, though, pointed out that adaptation literally means making something fit for a purpose. And the problem is that natural selection can't have a purpose because natural selection doesn't have a brain. The evolutionary process isn't trying to do anything because it doesn't have a goal. All that evolution has to work with is what there is. And evolution has no way of foreseeing what the future is going to be. So many evolutionary changes don't involve the evolution of anything radically new. What happens is that old structures that are already there get reused, like a boat gas tank getting reused as a Jeep gas tank, or a light switch getting reused as a dashboard switch for a fuel valve. This used to be called pre-adaptation. But evolution can't foresee anything, so we can't use the term pre-adaptation because that's not what evolution's doing. To replace it, Gould and Verba suggested the term exaptation. So what's an example? Here's an example on the molecular level that ties in with our example of evolution of the eye. Eye lenses have to be transparent, right? A brilliantly colored lens would kind of defeat the purpose. And to pass light, they have to be packed with water-soluble proteins that are more or less globular in shape, called crystallins. This is a bunch of different crystallins: alphas, betas, gammas, deltas, epsilons, and lambdas. These packed together in a very regular fashion in a quasi-crystal method, the way atoms in a crystal pack together in a very regular fashion. And that's what makes the lens transparent and what makes it refractive, what makes it not only pass light, but bend light, which of course is what you need a lens to do. The weird thing is that crystallins turn out to have very similar sequences to certain enzymes. And the really weird thing is that different animal groups with eyes have evolved lens crystallins from different enzymes. So in squid and octopus, I mentioned that these have extremely sophisticated eyes, easily on a par with your own. They've got lenses. Those lenses have crystallins. The crystallins work about as well as yours do, but the sequences aren't anything like yours. The sequences of squid lens crystallins are actually very similar to the sequences of a digestive enzyme called glutathione S-transferase. Weird. The lenses of scallops, very similar to a digestive enzyme called aldehyde dehydrogenase. Jellyfish, the lenses very similar to ADP ribohydroglycolase. Where vertebrates come from, I'm not sure we fully know, but they don't share much in common with squid crystallins. They come from a different source. And the idea is that this is tinkering on the molecular level. As eyes are evolving in sophistication, the idea is that a gene for an enzyme got recruited to do something different. This can happen because there's a number of ways in which you can have gene duplications. If you've got unequal crossing over in meiosis, you can have offspring that have two copies of a gene that have multiple copies of a gene instead of just one on each chromosome. They might have two on one chromosome. And when that happens, when you duplicate part of a chromosome, one of your gene copies might be available to evolve into something else. So you don't lose functionality. You don't lose the enzyme that you started with when you evolve lens crystalline. So, as eyes were developing, an extra copy of this enzyme may have been available. Cells in the eye began expressing it, and its enzymatic function isn't important. It doesn't really matter what the protein does. What matters is that it has a shape that packs together in a way that's conducive to transmitting and refracting light. That's exaptation for you. It doesn't really matter what the molecule is. What matters is that it's got the shape that's needed for a particular function at a particular time. And that's why groups with eyes have evolved such different lens crystallins. Give you another example, and this is one that was studied in real time. Um, there's an island off the coast of Croatia in the Mediterranean Sea called Podkopište. It's home to a species called the wall lizard, Podarcus sicula. 5 pairs of wall lizards were deliberately introduced from Podkopište to the nearby island of Podmurcharu. And Podmurcharu did not have any lizards on it at all at the time. So we start with a little founder population, right? This is a lot like what we've talked about. It's the founder effect. We'd probably expect to see that isolated population that we've just helped disperse to Podmurcharu become different from the population that it came from on Podkopište, right? This is plain old dispersal, probably leading to speciation. In particular, the Podkopište lizards were mostly feeding on insects. Podmurcharu, however, has a wider range of plant foods available. And when they came back and checked on the Podmurcharu lizards in 2004, so think it over, this is 33 years later, which might be 66 lizard generations later. I'm not sure how long a generation lasts, but probably several per year. So after about 66 odd generations, the Podmurcharu lizards, their diet, excuse me, consisted of up to 60% plants. On Podkopište, less than 10% of the lizard's diet was plants. So the Podmurcharu lizards had adapted to a different diet. As you might expect, this caused changes in the Podmurcharu lizards. The Podmurcharu lizards had bigger jaws, they had larger heads, and as was measured in the lab, they could exert greater bite force because it requires more force to bite into a leaf than it does to a nice, tasty maggot or fly or something like that. So there were physical changes that were noticed. There were behavioral changes. The Podmurcharu lizards were no longer territorial, probably because they had a food supply that was more even, not common at some parts of the year, but rare at other times, and probably more dependable. So there was much less to fight over. So the Podmurcharu lizards had lost their territoriality. They'd evolved physical changes to adapt to a new diet, and they had evolved, if you dissected them, they developed a little fold on the inside of the intestinal wall. There and right there. There's that little fold. It's called a cecal valve. And what it enabled them to do is close off part of their intestine and turn it into a fermentation chamber for plant foods. This was the secret to how they were surviving and thriving on a diet of 60% plants compared to their ancestors who'd been eating less than 10% of their diet as plants. They'd evolved the ability to ferment. And it's not that they evolved anything new. They took the existing lining of their guts and just folded it a little bit and added a little slip of muscle. So they're working with what's already there. Nothing radically new is getting added. But even adding a little thing that's put together from parts that were already on the shelf gives them this capability to digest a type of food that they couldn't digest before. So that's a new structure, and it only appears within 33 years. You may be saying, big deal. So a lizard evolves a slight fold of its guts. That's not a major evolutionary change. That's nothing. The thing is that major changes do not look major at the time that they happen. And I want to demonstrate this by looking at air breathing fish. All right, the fish that you know mostly respire with their gills, but many fish will attempt to gulp air if they're under oxygen stress. When I was a kid, we'd go to see my grandmother in Iowa, and her house was right across the street from the city park. And I'd just go over there and play, run around by myself all day. Come back when you're hungry, that kind of thing. I guess kids don't get to do that these days. And the park had a fountain in it, and there was a pool around the fountain, and the pool usually had goldfish. I'm not sure if the city put them there or, people who were tired of their pets put them there, but they were goldfish. And this being August, it was often pretty hot, and the water sometimes got kind of murky. And I'd see the goldfish coming up to the surface of the pond and doing this little gulp, gulp, or glub, glub thing. They were gulping air and trying to absorb whatever oxygen they could from the air that they gulp. And a lot of fish will do that if you put them under oxygen stress. If it happens in your home aquarium, it's a sign that you immediately need to clean the water and probably put in an air stone or something like that. It means your fish are in real trouble. So a lot of fish will do that, and they will try to absorb as much oxygen as they can um, through their gut. If you think about it, your gut is lined with mucous membrane. It's pretty good at absorbing substances anyway. So fish will try to get some oxygen by absorbing it from bubbles of air in their gut that they've swallowed. Here's the thing: fish that live permanently in habitats that are prone to oxygen starvation, many of them have evolved specialized little pockets and folds in the gut that increase the amount of oxygen they can absorb. And if you think about it, if you think about it from the point of view of a fish living in an area that in a population that gets oxygen stressed on the regular, any little advantage that you've got might be the difference between you having babies and you not having babies. Any little extra increase in the surface area in your gut and or in the vascularization, the number of blood vessels that you have available in the lining of your gut, any little increase in that, no matter how small, is going to provide some benefit. Any little extra pocket or pouch or fold is better than nothing. It will enable you to absorb more oxygen than your neighbors that don't have it. And it will give you that much better a chance of surviving and passing on your genes. And so we see this all over the fishy world, and different groups of fish have evolved this independently using different parts of their bodies. So the South American electric eel, this is the one that can cause a noticeable electric shock. Living in the Amazon basin as it does, it tends to be exposed to low oxygen conditions because its water can be very hot and full of decaying vegetation. And it gulps oxygen and it gulps air and absorbs the oxygen through its mouth. and the lining of its mouth is loaded with capillaries um, that absorb the oxygen. That's exaptation again. It's not the evolution of something new. It's taking things that are already there, a mouth with a mucous membrane with capillaries in it, and retooling it to do something that it couldn't do before. At the other end of the digestive tract, over on the left, that's a South American armored catfish in the genus Corydoras. These are popular as cleaner fish in freshwater aquariums. I kept a tank when I was a kid, and we always used to have a Corydoras. They're always rooting around on the bottom, snurfling up, particles of food that everybody missed. And there's an unrelated group of fish shown below. That's an Asian loach down at the bottom, genus Misgurnus. Both of these, even though they're not related to each other, they both will go to the surface, gulp a bubble of air, and absorb it through vascularized intestinal linings, not the mouth, but the intestine. They'll swallow bubbles of air and push them all the way back. I used to see my little Corydoras do this even when there was plenty of oxygen. He would just root around on the bottom and then just shoot to the surface, gulp a bubble of air, dash back down, and go on right with what he was doing. And they'll do this even when there's plenty of oxygen in the water. They still need to gulp air, absorb oxygen through their intestines, and then get rid of the rest of the gases, which means that these are among the few fish that fart. I'm serious. The waste gas that they can't absorb has to go somewhere. So it comes out the anus. It's farting fish. Aren't you happier knowing that? That there is such a thing in this world as fish that fart. Okay, another group of fish that I used to keep. Down on the bottom, that's a dwarf gourami. Gouramis and bettas, aka Siamese fighting fish. These are the fish that you see at PetSmart being sold in little cups. Most fish would die very quickly if you put them in tiny cups. But bettas and gouramis belong to a group called the anabantoids. And branching off the gill chamber, they have this very complicated folded chamber in the head. The folds give it this very kind of windy shape to it. It's called the labyrinth organ because in cross section it looks a little like a maze. Those maze-like folds branching off the gill chamber give extra surface area and allows the fish to gulp air and absorb oxygen from it in the labyrinth. That's how bettas can survive. They're not super happy, but they can survive in the pet store in tiny glasses of water where most fish would die pretty quickly if you put them in conditions like that. Now, if you own one, you really do need to give it more space. It's not going to be happy if you keep it in a wine glass, but they can live in very oxygen deprived water in very small amounts of water because they can gulp air and absorb it in the labyrinth organ inside the head. Completely convergently, snakeheads have been in the news. That's an Asian fish that's been introduced to Arkansas. That's a fish that's kind of an environmental problem, as the Adamses can tell you. They've actually done work on snakehead eradication because a snakehead can actually crawl over land, crawl into your pond, eat everything in it, and then crawl out and find another pond. Snakeheads can pull that off because they've got air absorbing chambers branching from the gills. And so does this fish. This is the Indian stinging catfish. You can see it's got really long barbels right there and a long snaky body. And there's its head, there's the gill chamber. And running back from the gill chamber, you have this elongated air sac. So a stinging catfish can gulp air. push it back into that air sac and absorb oxygen from there into the bloodstream. Yet more South American small catfish, like little Otocinclus right here. There's the esophagus, so food comes in there. There's the stomach right there. Food passes through the pyloric valve into the intestine, which comes down right there. But the stomach has this huge pouch branching off the stomach called the diverticulum. And that's for absorbing oxygen from swallowed air. So there's a little stomach here that actually digests food, and a huge diverticulum pouch off the stomach that's used to absorb swallowed air. So first of all of these are examples of exaptation. They're not really evolving anything new. They're taking what's already present. Um, you know, this this diverticulum is basically just an expansion of part of the stomach. It's an extra pouch, but it evolves from the stomach. It's made of the same stuff as the stomach. In a related fish called Scoloplax, the stomach itself is mostly used to absorb oxygen. There's a tiny strip that still secretes digestive enzymes, but most of what the stomach is doing is absorbing oxygen. So you're taking an existing structure and using it to do something completely different. Here, you're taking the gill chamber and just making part of it extend, making part of it grow bigger. Same thing with the anabantoids. You're just taking the gill chamber and expanding a portion of it and folding a portion of it. But in all of these cases that we've seen, you're building on something that's already there. So it should be easy to see how an early ancestor of all bony fish actually evolved a pair of simple pouches that branch off its esophagus. This is one of the earliest branches of bony fish that we still have. This is Polypterus, the beach ear. And there's a live beach ear over there. And if you look at a CAT scan of a beach ear, because somebody did that, highlighted in yellow, you've got a pair of pouches that branch off the esophagus. And their use is to absorb oxygen from the air. And this is exaptation because the fish already has an esophagus. You're just taking what's already there and kind of stretching it a bit and adding some pouches, increasing the surface area that's available for absorbing oxygen. But you're building on structures and capacities that all fish already have. Okay, that pouch, there's Polypterus right there. E, that's the beach ear. And you can see there's the esophagus, and there you have this pair of pouches coming off of it. That's E right there. That's what bony fish basically start out with. It's retained in sturgeon, for example. You can see there's the esophagus, this. Food passes through there. And then you have this, there's the esophagus. And there you have this air chamber that's connected to the esophagus by a little opening right there. That air chamber has a folded wall in Lepidosteus and Amia. That's gars and bowfins. They've got this air chamber, which is what enables gars and bowfins to live in very oxygen-deprived water. Bowfins can live where almost nothing else can. I think Erythrinus is a lungfish. I know Epiceratodus is. They've got the same thing. They've got these pouches branching off the esophagus that are used to absorb atmospheric oxygen. And lungfish, incidentally, are so dependent on atmospheric oxygen that they are among the only fish that can drown if you keep them away from the atmosphere. They've got gills, they use them, but they cannot completely fulfill their need for oxygen using only their gills. They have to gulp air. And here's the thing, we see the same thing in tetrapods down here at the bottom. That's land vertebrates. That includes you. Your lungs began when you were a tiny embryo as little pouches coming off your esophagus. Now, don't read too much into this, but that's how lungs evolved, is very early lineages of fish developing pouches on their esophagus that were useful for absorbing air. This is the way that yours develop. This is the way that what yours looked like when you were just a few weeks post-conception. And this also happens to be the way that they evolve. They're little extra pouches on the esophagus for absorbing atmospheric oxygen. You've gotten very, very good at this. Your lungs have evolved all of this branching and all of these air sacs that add enormous amounts of surface area because you're very good at absorbing atmospheric oxygen. And your lungs have become very highly specialized, but your lungs weren't always that complex. You know, in amphibians like frogs, some of them don't even have lungs. The ones that do, they're basically just bags. And your very distant fishy ancestors had some kind of little pouch branching off the esophagus. And it started out as no more complicated than that little fold in the guts of the Podmurcharu lizards. You know, just a little extra surface area, just a little fold, just a little pouch, just a little expansion, just, you know, something that gives you a slight edge. And what's happened since has basically been working out potentials that were already present, possibilities that are already there, taking this little pouch and in many lineages, adapting it to different lifestyles. For example, in most ray-finned fish, let me go back one. In some bony fish that you know, herring being an example, this pouch is called the swim bladder, and herring, as well as sturgeon, still retain a connection between the esophagus and the swim bladder. That's called being physostomus. They've still got a, they can still gulp air and pass it into that bladder. You can still gulp air and pass that into your swim bladder, but in most ray-fin fish, that pouch pinches off completely. It's called being physoclistus. I won't test you on that, but you can impress Reed Adams if you ever take one of his courses. That pouch pinches off its connection and becomes just completely self-contained. The fish can now secrete gas into it or take gas out just using its circulatory system. And that swim bladder becomes this gas bladder that controls the fish's buoyancy. It's no longer able to absorb oxygen. It's no longer connected to the outside world. It still is able to absorb gas. Well, it still is able to pass gas into the circulatory system or receive gas from the circulatory system, the way that your lungs pass oxygen into your circulatory system and receive carbon dioxide from it. But the fish in this case uses that not to breathe, but to control buoyancy. Controlling the amount of gas in the swim bladder determines at what level the fish rises, floats in the water, whether it rises or sinks. So you've got lots of exaptation going on there. You started with a dinky little pouch off the esophagus way back when you were a fish, and that pouch increased surface area available for absorbing oxygen. In the line leading to you, your pouch has become bigger and bigger and bigger and more branched and folded and complicated. But that's all exaptation. That's modifications added to something that was already there. In most bony fish, that connection with the outside has been lost, and the ability of that pouch to um, uh, to absorb and contain gases has gotten reused again to create an organ that controls buoyancy, instead of an organ that absorbs oxygen for respiration. So you've got multiple instances of exaptation, not building anything new, but working with potentials that you've already got. And that is an extremely common way that evolution works to build complex structures. Um, and I don't have time to get into all of this, but if you really want the full story, you ought to take comparative vertebrate anatomy with Dr. Cash, and you'll get all of this in spades. This is a beautiful case of exaptation. Bony fish have arteries that run through the gills, right? The heart pumps blood through the gills, and that's where the blood picks up oxygen. And the heart's been removed here, but blood would do this, pass through the gills, and then pass to all parts of the fish's body. Okay. What's happened in the evolution of land mammals or land vertebrates, not just mammals, is that those gill arches have gotten rerouted. And one particular branch of them, the arteries and veins of the sixth gill arch, end up forming the pulmonary circulation. What used to be the fourth arch, what used to be the fourth gill arch's arteries, becomes your aortic arch, right? That's your aorta. You've heard of that, right? That's the main blood vessel, main artery that leaves your heart and says branches to your arms and to your head. All right, that's your fourth gill arch, or it was when you were a fish. And the arteries and veins of the sixth arch get rerouted a little bit, and they give rise to the pulmonary arteries that carry blood to the lungs, where it gets oxygenated. They also give rise to a little connection called the ductus arteriosus, which is supposed to close right about the time of birth. If it doesn't, the baby may have much less efficient respiration. They used to call these blue babies because they couldn't oxygenate their blood properly if the ductus arteriosus didn't shut. But that ductus arteriosus right there is just another remnant of what used to be the artery of the sixth gill arch. So the arteries, excuse me, that used to carry blood to the gills, some of them have been lost, others of them have been rerouted a bit. And what you end up with is the pulmonary circuit, the way that the heart pumps blood to your lungs, back from the lungs with oxygen in it, and then out to all parts of the body. So you've got that double loop, heart to lungs to heart and heart to body to heart. And that's all an example of tinkering or exaptation. You could call it both in this case, taking a structure that's already there and rerouting it to do something that's quite different. So something as complicated as your pulmonary circulation and your lungs is an example of several exaptations of structures that were already present in very early bony fish, something on the order of 420 million years ago. Okay, this is where the jaws are supposed to hit the floor. I'm always impressed with this. And again, if you want the details of exactly how this was done, go take comparative vert anatomy. All right, I've got 11:02. Let's reconvene at I'm feeling generous. I'm going to stop the recording.